Welcome to Chapter 4, Biosensor and Drug Delivery Material Design. This is the Applications chapter of our Bioinformatics-Introduction series. We'll explore real-world applications of the computational skills you've learned, from biosensor design to drug delivery systems, and discuss career paths in this exciting field. By the end of this chapter, you'll understand biosensor design principles, including recognition elements and signal transduction. Explain drug delivery system design strategies using nanoparticles and liposomes. Design peptide materials with self-assembly and functional capabilities. Understand career paths in biomaterial and pharmaceutical companies, and develop a learning roadmap as a bioinformatician. Biosensors are devices that use biomolecules to detect specific target substances. The signal flow goes from the target analyte to a recognition element or bioreceptor, which specifically binds the target. This binding event is converted to a measurable signal by a transducer, amplified by a detector, and finally output as a quantifiable signal. The three major biosensor components each have multiple options. Recognition elements include antibodies for high specificity, aptamers which are synthetic DNA or RNA, enzymes for catalytic detection, and DNA for nucleic acid targets. Transducers can be optical using fluorescence or absorbance, electrochemical measuring current or potential, or piezoelectric detecting mass changes. Sensor sensitivity depends heavily on the binding affinity of the recognition element. This Langmuir binding curve shows how lower dissociation constant Kd leads to higher sensitivity. Antibody A with Kd of 1 nanomolar has a limit of detection around 0.18 nanomolar. Antibody C with Kd of 10 picomolar achieves LOD of just 0.0018 nanomolar, 100 times more sensitive. Aptamers are synthetic nucleic acids that can bind targets with antibody-like affinity. We can computationally design aptamer candidates by generating random sequences and predicting their stability. Key features include GC content for thermal stability and G-quadruplex motifs for structural stability. In practice, SCLEX experimentally selects the best binders from random libraries. Drug delivery systems efficiently transport drugs to target tissues. DDS design involves three key decisions: carrier selection from options like liposomes, polymer micelles, or nanoparticles, drug encapsulation strategy, and targeting approach, either passive through the EPR effect in tumors or active using surface ligands for specific cell receptors. Drug release profiles determine therapeutic efficacy. This plot shows cumulative release over 48 hours for 3 formulations. Sustained release with low burst and slow rate maintains therapeutic levels longest. Rapid release reaches high concentrations quickly, but may exceed the safe maximum. The goal is keeping drug levels within the therapeutic window between minimum effective and maximum safe concentrations. Targeting peptides direct drug carriers to specific cells. RGD tripeptide binds integrins overexpressed on tumor cells. Folate receptors are normally targeted by attaching folic acid itself to the carrier, rather than by a peptide motif. Transferrin receptor ligands enable brain delivery. We design targeting peptides by combining core motifs with flexible glycine-serine linkers, then evaluate physicochemical properties like charge and hydrophobicity. Self-assembling peptides form hydrogels useful for tissue engineering and drug delivery. We predict assembly tendency using beta sheet propensity scores for each amino acid and amphiphilicity from alternating hydrophobic hydrophilic patterns. The peptides RADA16 and MAX1 are proven self-assembling sequences. Random sequences lacking these features won't assemble. The self-assembly process proceeds hierarchically. Individual peptide molecules with alternating charge patterns first form beta sheets through hydrogen bonding. These sheets stack into nanofibers, which entangle to create a 3D hydrogel network. This biocompatible scaffold can encapsulate cells for tissue engineering or drugs for controlled release. Antimicrobial peptides kill bacteria by disrupting their membranes. Effective design requires positive charges from lysine and arginine to bind the negatively charged bacterial membrane, plus hydrophobic residues like leucine, phenylalanine, and tryptophan to insert into the lipid bilayer. The amphipathic structure with charged and hydrophobic faces enables membrane disruption. We can generate antimicrobial peptide candidates computationally using alternating charged and hydrophobic residues. Each candidate is evaluated for positive charge count, hydrophobic residue ratio, and predicted activity. However, computational design must be validated experimentally through cytotoxicity testing to ensure safety, minimum inhibitory concentration measurement for efficacy, and protease stability assessment. Let's understand the pharmaceutical development workflow. Stage 1, basic research, takes 6 to 12 months for target identification and docking simulations, producing 100 to 1,000 candidates. Stage 2, in vitro evaluation, runs 12 to 18 months measuring binding and toxicity, narrowing to 10 to 50 hits. Stage 3, in vivo studies, takes 18 to 24 months to select 1 to 5 leads. Stage 4 is the clinical trial program itself, phases 1 through 3 in the regulatory sense, and that takes 3 to 7 years to reach market. Note the wording. These development stages are not the same thing as the numbered clinical phases. Drug development is expensive and risky. Costs escalate by orders of magnitude across the stages, from the hundreds of thousands of dollars typical of early basic research up to the hundreds of millions spent on clinical trials, with industry estimates putting the total cost of bringing one drug to market at roughly $1 to $2.5 billion. Success rates plummet. 30% of projects advance from Phase 1, 20% from Phase 2, 10% from Phase 3, and only 5% of clinical candidates reach the market. The cumulative probability of a basic research project becoming a marketed drug is just 0.03%. Career Path 1 is in biomaterial companies like Terumo, Olympus, Fujifilm, and Curare. Roles include biosensor development, medical device design, and regenerative medicine research. Annual salaries fall broadly in the range of $50,000 to $120,000 depending on experience and country. Treat these as rough orders of magnitude, not quotes. Required skills include bioinformatics, materials science, and project management. Career Path 2 is in pharmaceutical companies like Takeda, Astellas, Daiichi Sankyo, and Chugai. Roles focus on antibody drug design, nano-drug delivery development, and targeted therapy optimization. Salaries are higher at $60,000 to $150,000. You'll need protein engineering skills, molecular docking expertise, and pharmacokinetics knowledge. Career Path 3 is in biotech ventures like Spiber for structural proteins, Euglena for microalgae, and PeptiDream for special peptides. Roles involve novel biomaterial creation, computational design, and potentially founding your own company. Base salary is $40,000 to $100,000 plus stock options. Success can bring 10x or greater returns. This path requires entrepreneurial spirit. Here's a 3-month plan for building your foundation. Month 1, complete this bioinformatics series, practice BioPython coding, learn PyMol visualization. Month 2, master Autodoc Vena docking, learn machine learning basics with scikit-learn, create your GitHub portfolio. Month 3, execute an original project with your own data, write blog articles to share your work, and participate in conferences or study groups. For longer-term planning, in 1 year, spend Q1 on fundamentals and Python implementation, Q2 on machine learning and data analysis, Q3 on a corporate internship, and Q4 presenting a poster at a conference. Over 3 years, Year 1 builds basics and implementation experience, Year 2 develops original research and new methods, and Year 3 focuses on paper publication and job hunting. Let's summarize Chapter 4. We covered biosensors including recognition elements, signal transduction, and sensitivity optimization. Drug delivery systems with nanoparticle carriers, targeting ligands, and release control. Peptide materials including self-assembling peptides and antimicrobial peptide design. And finally, career paths in biomaterial companies, pharmaceutical companies, and biotech ventures. That wraps up the applications chapter. So far, you've learned protein structure analysis, sequence-based machine learning, molecular docking, and biomaterial design. One chapter remains. In Chapter 5, we'll tell the story of AlphaFold, the AI system that cracked protein structure prediction, and you'll learn to download, score, and analyze its predictions yourself. After that, consider the bioinformatics-introduction for small molecule analysis, or data-driven materials design for broader materials informatics. Exercise 1 asks you to propose 3 strategies for improving biosensor sensitivity. Think about the 3 components we discussed. Hint: consider improving recognition element affinity, amplifying the transduced signal, or reducing background noise. Each strategy has multiple implementation approaches. Exercise 2 asks you to compare passive and active targeting in DDS design. Passive targeting relies on the EPR effect where tumor vasculature is leaky. Active targeting uses ligands for specific receptors. Consider the advantages and disadvantages of each: cost, selectivity, intersubject variability, and immunogenicity. Exercise 3 is challenging. Design a self-assembling peptide hydrogel meeting specific criteria. It should be 12 to 16 amino acids long, have high beta sheet forming ability, be amphipathic, and include the RGD cell adhesion motif. Apply the design principles we learned: beta sheet propensity, alternating patterns, and functional motif placement. For further reading, we recommend 3 landmark papers: Langer 1998 in Nature on drug delivery and targeting, Zhang 2003 in Nature Biotechnology on molecular self-assembly for biomaterials, Zasloff 2002 in Nature on antimicrobial peptides. These foundational works will deepen your understanding of the field. Thank you for completing this series.